This is my presentation on the implications for the survival of frogs caused by ranaviral infections. Ranavirus is found in a wide array of amphibian species and is present globally, but this presentation focuses predominantly on ranavirus within frogs and toads in the UK. Other species and continents are mentioned for comparisons and examples, but this is the main focus. We originally got an understanding of what ranavirus is due to frogs dying seemingly randomly in the 80s and 90s, showing a variety of symptoms and sometimes seen with secondary bacterial infections. Since it only started being reported in the 80s, this is an indication of it being a relatively new disease here in the UK, which was most likely spread here from the commercial importation of goldfish and bullfrogs from North America. It is only active in warmer temperatures, so if frogs are found with symptoms or dead outside of the summer months, then it is more likely that there was an outbreak of some other kind of infection, or perhaps they died of natural causes. The clearest sign that a frog population is suffering from ranaviral infections would be mass mortalities of adult frogs in the summer in or around ponds, especially if they have been thin or lethargic a couple of days before. So carrying on, ranaviruses have been reported on 5 different continents. They belong to Iridoviridae, a family of viruses that use amphibians, fish and arthropods as their natural hosts, and this is known to cause morbidity and mortality in these animals. Anthropogenic spreading, which includes methods like infected animals being used as baits or recreational equipment that has been within an infected area in use, as well as the more obvious import and export of infective animals for food or in the pet trade, has been the main cause for unaffected and naive populations to be exposed to this infection, and so leads to their subsequent mortalities. The necrosis that ranavirus causes in these wild amphibians leads to their organs failing and most commonly death. The clinical signs include swellings, ulcers and many other easy to spot symptoms that are looked at in the next slide. So symptoms of ranavirus. It can be difficult to diagnose ranavirus in animals without the use of a post-mortem, but they can display one or more of these symptoms that are listed. So lethargy, emaciation, arrhythmia, ulceration, bleeding, specifically from the mouth and anus, limb necrosis and eye problems. The 2 main symptoms of ranaviral infection in frogs are the skin ulcerations and internal bleeding. The ulcers, they're generally easy to spot, especially if they are formed on the underside of the frog's pelvic region and on their hind limbs. So sometimes the ulceration can cause a loss of digits if it's quite severe, and you can see that in the top right-hand image of figure 1. The bleeding can be a bit trickier to spot, but is evident from the mouth or cloaca as the underside of the individual starts to redden. The reddening of the frogs has led to the ranavirus being nicknamed the red leg disease, which is a fairly misleading name for this disease, as reddened legs can be symptoms of other types of infection, and frogs are suffering from ranavirus might not actually exhibit this symptom. But unfortunately, the most common symptom is the mass mortality in an individual population, and by which case the diagnosis is far too late for that group. So the spread of ranavirus. Ranavirus was originally found in the southeast of England, but has spread all across the UK, being reported in Cardiff, Lancashire and Newcastle, to name a few places. Around the country there are certain hotspots of ranavirus, but it is unknown how exactly this has happened. So if you look at the figure on this slide, this is from a study in 2017 of the UK distribution of ranavirus in frogs, and it's mostly clumped down in the southeast of England, which is where it started, but it's spread all the way up the country, up to the north of Scotland, as you can see. So as mentioned before, the original introduction of the disease in the UK has been suggested to be caused by the importation of amphibian species and fish from North America, but just how it moved around the UK is presently a mystery. There have been studies that say the spread of ranavirus from population to population is 80% due to frogs just moving of their own accord, but 20% due to human impacts. But it is thought that the main reason for this spreading is the abundance and crowding of the frogs in their populations. So it is known that the disease is temperature and density dependent, peaking in the summer months when the temperature is particularly high, and also when there is a large number of frogs in and around ponds. This is how the disease thrives, and this is how it spreads. Due to it still being unclear how exactly the disease disperses, it has been recommended that introducing plants and animals, including fish, into your own pond should be avoided, as it could be detrimental to the current amphibian population habituated in there. When looking at amphibian conservation in general, we generally go to the IUCN, or the International Union for the Conservation of Nature. And as they say, amphibians are considered to be the most threatened vertebrate class in the world. There have been many global declines and extinctions that have affected amphibians, including habitat loss, fungi, which most commonly is the citrid fungus, and of course disease and infection. Amongst these factors, several types that are found in the genus Ranavirus have been linked to the majority of amphibian mass mortality events that have been reported. One study by Mutz et al. In 2006 found that 43% of all reported die-offs in the USA from 2000 to 2005 were caused by ranaviral infections. Comparably to this, Green et al. In 2002 reported that 57% of the mortality events, which were investigated by the US Geological Survey National Wildlife Health Centre from 1996 to 2001, were also caused by ranavirus. So it is a big impact on the survival of amphibians. So as ranavirus causes mass mortalities in the amphibian populations, they drastically affect the structure of the populations that they infect, and also the likelihood of that species' persistence and survival. This threat is particularly high for the less abundant species of amphibians, as they will find it harder to recover after mass impermanence events. Mortality events in amphibians have been reported worldwide due to ranaviral infections. Ranaviruses have also been isolated from wild-captured amphibians in Australia, but the die-offs in the wild from these infections are still unknown on this continent. Despite all these statistics and impacts that have come from ranaviral infections, it is still debated whether or not these infections present a significant threat to the biodiversity and conservation of amphibians worldwide, according to the IUCN. So the treatments, there's not that much info on this slide because currently there is not a treatment in place that cures ranavirus in amphibians. It is proposed just instead that these positively tested animals are put into isolation in order to prevent other animals from getting infected. This isolation will also provide optimal husbandry conditions for the amphibian and will also expose them to much less stress, which gives the individuals the best chance of recovering and developing a resistance to the infection. So rather than curing these infections, the best measures to take are preventative ones to stop it from spreading further. But speaking of stress and conditions, stressors. External stimuli can be a natural stressor for ranavirus in frogs. These stimuli include changes in the ambient temperature, limitation of food, or the threat of predation. Water temperature change is one of the most common environmental stressors, and it can impact the susceptibility of the infection. A study by Cooper et al. In 1992 found that cold-induced immunosuppression has been demonstrated in multiple species of frogs and toads. Thus far, no studies have been published on the impact of predation or interspecies competition relating to resource limitation on their susceptibility of amphibian hosts and ranaviruses. Another example of a natural stressor for amphibians includes breeding. Ranaviruses generally are not as pathogenic to adults as larvae, but periods of stress as an adult may cause chronic infection. The origin of some stimuli that act as stressors are sometimes anthropogenic, meaning that they are from human-influenced origin, and it includes stimulants such as the water quality within agricultural operations. So Carey et al. In 1999 hypothesised that an increase in the number and intensity of these anthropogenic stressors are partly, if not more, responsible for the emergence of wildlife diseases. Amphibians are especially susceptible to these stressors in the environment as they have semi-permeable skin, which means that they uptake toxins. So in environments such as farms, the agricultural practices and fertilisers, they may directly decrease the water quality within the wetlands through adding nitrogenous compounds, and the agricultural land use may indirectly compromise amphibian immunity whilst increasing the chances of susceptibility. Another common anthropogenic stressor is the use of pesticides, which negatively affect the immune systems of amphibians, which potentially drives the emergence of ranaviral infections. Pesticides can be applied to aquatic systems through either the direct application of them, terrestrial runoff, and also through windborne drift. Pesticides may also trigger environmental stimuli due to their ability to change food web structure, which applies itself as a natural stressor and increases the susceptibility. It is clear that pesticides do affect the immune function of amphibians, but more research is required to clarify these impacts and their link to the emergence of ranavirus in these anurans. Any stressors can increase the chances of pathogen infection and morbidity because of the reduced immune function. So, as on the treatment slide said, if an animal is tested positive, it is best to isolate them and make sure they are in optimal conditions so these stressors do not provide the ranavirus with more fuel to essentially bring down the individuals. So human impacts, one of the most common stressors for these animals. Human impacts can drastically affect inuran populations. Habitat fragmentation is the most common threat to these animals caused directly by humans, according to the IUCN, and it leads to the isolation of populations both demographically and genetically. It has been suggested by Altzier et al. In 2003 that the increased occurrence of inbreeding in isolated populations leads to the loss of genetic heterozygosity, which then can potentially increase pathogen infection and rates of morbidity. Pearman and Garner in 2005 gathered evidence that the populations of genetically isolated Ranaviruses had a far greater chance of experiencing mass mortalities from the introduction of ranavirus. As a direct consequence of habitat fragmentation, these results suggest that inbreeding and genetic isolation increase the likelihood of a ranavirus epizootic occurring. Although when inbreeding occurs as a consequence of directional selection associated with the ranavirus die-off, it may provide advantageously, according to Teacher et al. In 2009. The increased probability of being in contact with an infected individual is a direct result of habitat fragmentation as well. There have been several studies published that document an increased nestedness and increased relative abundance of some amphibian species that inhabit breeding sites situated within agricultural landscapes. Pathogen pollution was a term coined by Cunningham et al. In 2003, and it refers to the possible transportation and introduction of novel ranaviruses by humans into naive populations. It is suspected that the introduction of these novel ranaviruses are probably a greater risk than the endemic types, as studies have demonstrated that they are far more pathogenic than the actual ranavirus infections. So, conservation strategies. At present, there is a very limited amount of studies that have been conducted on the use of conservation strategies to reduce disease emergence. Even though ranaviral infections may emerge because of the aforementioned natural stresses, it also emerges due to the anthropogenic stresses that humans cause them. It is therefore key that we put strategies in place that reduce the impact of anthropogenic impacts. As well, if we introduce strategies that reduce human activity, this will limit the establishment of any novel viruses. It has been recommended by Semlich and Bodie in 2003 that we establish undisturbed vegetation buffers that go around breeding sites of frogs, as this helps to reduce the human impacts on the aquatic environment. They suggested having a buffer of at least 30 metres of vegetation to hedge this environment and protect the amphibians within it. Adding to this strategy, Gray et al. In 2007 documented that the exclusion of cattle farming and electric fencing from wetlands seems to reduce the prevalence of ranavirus in a few amphibian species. So to combine this study with that of Semlich and Bodie, cattle should be farmed around 20 to 200 metres from any amphibian breeding sites, and then add a 20-metre buffer to reduce any of the effects of the cattle and also of humans, which then prevents the emergence of the infection. More simpler strategies would be to simply restrict chemical applications to days when there is less wind, and this will reduce the amount of pesticides being transported into frog breeding sites. A slightly different approach, I would say, is the strategy of educating the public. Now, I feel personally that this is one of the most effective ways in conservation to prevent something, because you are aiding people who will have these ponds with frogs in their garden, and they will know what to do or who to call or what to look out for, and so acts as a safeguarding measure to protect these animals and stop the spread of this infection. The Zoological Society of London and Stephen Fry recently made a video within the last year or so that provides information on what ranavirus is, how to spot it, and what to do if you do spot it. And I feel like this is a great way to try and save these species. So look it up. And so to conclude, ranaviral infections impact populations of common frogs and toads drastically. As we've seen, it causes mass mortalities, as that is the main symptoms. So symptoms, ulceration, bleeding, and death. The first two, very easy to spot, and so once spotted, you can do something about it. The last one, also easy to spot, but it's a bit too late then to try and help, other than stop it spreading from outside of that pond. There is currently no cure or treatment for the disease, so the best measure to take are preventative measures. Be cautious, isolate these individuals so the rest of the animals are okay. So So external anthropogenic stimuli act as stressors. So like we said, in the environment, any changes in temperature, water quality, anything to do with humans, affecting them, just in contact with other infected animals, there are a big list of stressors and ways that they can contract these diseases. And so we need to be able to cut them down. So the main strategies involve reducing the effect of humans. As that is probably the easiest thing for us to sort out, as opposed to the environment, which we don't have that much control over, we need to impact differently on these ponds as humans. We need to be careful with where we put our pesticides and when we put our pesticides down. We need to pay more attention to what we're introducing into our habitats in our garden. We need to make sure that there are not plants and fish that have been in contact with infected individuals being put within an uninfected population of frogs and toads around our areas, and that will reduce the spread and so will help to effectively stop this disease killing many more. Yeah, so that's everything. So to visit the Amphibian and Reptile Conservation Trust, and you can learn more about these threats. You can find out how you can help, you can donate, you can be a member. Yeah, just look after the frogs. Thank you.